Welcome to Medrock Medical Lectures and welcome to our one video lectures. Today's topic will be cell membrane and I am Dr. JK.
So let's get right into today's topic, cell membrane. In today's class, we'll discuss the various cell membrane models, the composition of cell membrane, and the factors influencing the fluidity of cell membrane, and the extensively studied membrane, that is the RBC membrane. The various models of cell membrane that were proposed. The first model was proposed by Daniel and Davison, and they gave the term sandwich model because lipids were sandwiched between two layers of proteins. The next model was given by Mr. Robertson, which was an extension of the previous model, and he gave the name unit membrane model. These models were discredited. Now, in 1972, two scientists, Mr. Singer and Mr. Nicholson, proposed the fluid mosaic model. Fluid because the cell membrane is not a concrete or rigid structure, and mosaic because it is made up of many smaller substances like proteins, carbohydrates, lipids. This entire cell membrane is a design that is made up of multiple small separate units. That's why they called it as mosaic, the fluid mosaic model. The composition of cell membrane. The major constituents are lipids, proteins, and carbohydrates. You have the extracellular fluid here and the intracellular fluid on this side. Lipids are arranged in two layers, that's why they call it as a lipid bilayer. And you have proteins that can either be an integral protein or peripheral protein. Peripheral protein can either be on both sides also, ECF or ICF. And you have certain small structures present on the proteins dangling in the ECF. They are your carbohydrates. So this is a cell membrane entire structure. Now coming to the first constituent, lipids. Lipids constitute 40% of the cell membrane. And now we'll look at the major lipid composition, which is phospholipid. Phospholipid contains a hydrophilic phosphate head as well as hydrophobic fatty acid tails. These phospholipids are arranged in two layers with the tails facing each other, which you saw in the previous picture. Now, why is this arrangement? Yes, that's me there. Now, you have the hydrophobic phosphate tail on one layer on this side and the other layer on this side facing each other. And these are the hydrophilic heads on one side and you have the hydrophilic heads on other. Now, why is this peculiar arrangement with the tails facing each other? You have the ECF and ICF on both sides here. And you know, ECF and ICF are aqueous compartments, that the major composition of ECF and ICF is water. So since tails are hydrophobic, they will be facing away from the water containing ECF or ICF, whereas the heads are hydrophilic, so they will face towards the water containing compartments. That is why this beautiful arrangement of lipids, and this arrangement is called as the lipid bilayer. The next lipid is cholesterol. Cholesterols are big molecules that are fused in between the phospholipids. They play a major role in controlling the fluidity of cell membrane, which we'll see later on in the lecture. The next composition is protein. Proteins constitute around 50 to 55% of the cell membrane. To understand proteins, you should first know about lipid rafts and the different types of proteins, the integral and peripheral proteins. What are lipid rafts? Look at this picture here. You have the bilayer, lipid bilayer here, and in the bilayer there is a special portion called as lipid rafts. What are lipid rafts? They are places in which the fatty acids or phospholipids are very closely packed with each other, because the fatty acids are all saturated and they contain high amounts of cholesterol also. These yellow, white structures here are cholesterol. So this makes this particular portion of the cell membrane a little rigid when compared with the other layers. Because it is a little tight, to allow particles to come into or go out of the cell, there are special proteins that are present here. Please note that proteins are not only present in the lipid rafts, but they are majorly present in the lipid rafts. The major proteins here are signaling proteins. Alright. So what are the types of proteins that are present, which we saw earlier before? The first one is integral proteins. Integral proteins transfuse the cell membrane. They either pass halfway or 3/4 of the cell membrane, or some proteins they transfuse the entirety of the cell membrane. If it crosses the entire cell membrane, they are called as transmembrane integral proteins. Whereas proteins that are present on the periphery, either on the ECF or ICF, they are called as peripheral proteins. And these proteins are anchored by special receptors called as GPI receptors. So what are the functions of membrane proteins? The main function is they help to transport substance either into the cell or out of the cell. They act as receptors, some act as enzymes, and some also play a role in immunity by giving antigenicity to certain substances. We'll look into the depth of how proteins help us transport us. So these are two transporters. This transporter is called as channels, and this transporter here is called as carrier protein. Now let's look at this example. As I said, this is called as a channel. Channels have a central hollow canal through which the substance can either come into the cell or go out of the cell. These proteins are called as carrier proteins, in which the substance attaches to one side of the protein and the protein changes its configuration or shape and transports the substance into the cell or out of the cell. These are called as carrier proteins. There is one more protein which is called as a pump or pump proteins. They are carrier proteins, but they transport the substance against their concentration gradient, utilizing energy. That's the difference between pump proteins and ordinary carrier proteins. Now coming to the last constituent, carbohydrates. Membrane carbohydrates occupy almost 3% of the cell membrane. The carbohydrates can either be lipids, glycolipids, or proteins, which are glycoproteins. The carbohydrates are called as glycocalyxes. The main functions of membrane carbohydrates. The carbohydrates are negatively charged particles. So they help to repel other negatively charged particles. As I mentioned, the glycocalyxes are negatively charged. So they do not let other negatively charged particles to enter into the cell. Another function is they provide glycocalyx to glycocalyx attachment. If you have two cells and there are two carbohydrates on the ECF, sometimes the glycocalyx of one particle will attach to the glycocalyx of the other. This plays a major role in few steps of blood coagulation as well as phagocytosis. Similar to proteins, some carbohydrates act as receptors as well as they also provide antigenicity. Now the next part of the lecture is about fluidity of cell membrane. The main factors that determine the fluidity of cell membrane are temperature, fatty acid saturation, and cholesterol. Increase temperature is going to increase the fluidity of cell membrane, because increased temperature will loosen all the lipids or fatty acids, which will increase the fluidity, and increased saturation of fatty acids is going to decrease the fluidity, because saturated fatty acids will be more tightly packed, so that will decrease the fluidity. So these two are vice versa also. Now cholesterol has a dual role. At increased temperature, it decreases the fluidity, whereas at decreased temperature, it's going to increase the fluidity of cell membrane. Now why is that? Now the first point I said, increased temperature will increase fluidity because it loosens all the fatty acids or phospholipids. Now cholesterol is present between the phospholipids. Now because increased temperature is pushing all the phospholipids away from each other, in high temperature what cholesterol will do is it will try to bring all the phospholipids together, thereby decreasing the fluidity. And the same the opposite happens in decreased temperature. When temperature decreases, it pulls all the fatty acids together, thereby it will try to decrease the fluidity, but at the same time cholesterol will push all the fatty acids away from each other, thereby increasing the fluidity at low temperature. So cholesterol alone has a dual role depending on the temperature and thereby affecting the fluidity correspondingly. Now the last part of the lecture, RBC membrane. The red blood cell membrane. The RBC membrane was extensively studied and a lot of proteins were discovered. The integral proteins that you all must know are glycophorin and band3 protein, and peripheral proteins are spectrin and ankyrin, to name a few. Just know the names of these proteins for now. When we study later about spherocytosis, elliptocytosis, and other RBC membrane defects, you will know the importance of these proteins. So with that, we come to the end of today's lecture, cell membrane.
Now that the class is over, let me give you a homework. Find out if the phospholipids on the ECF side of the membrane and the phospholipids on the ICF side of the membrane are same or different. If they are different, let me know in the comments what is the difference. Remember that the factors that affect the fluidity, the different constituents of the cell membrane, all these make the cell membrane semi-permeable, which means that the cell membrane allows certain substances to pass while it inhibits the other substances. We specifically made this video because this is a prequel for our next big lecture series. If you could guess what the next series is, let us know in the comments. As always, please like, share and comment your suggestions. Please spread our videos to your friends also. Please subscribe to our channel. Your support, it means a lot to us. See you soon in the next lecture.